Hello, thank you for joining me today. I'm Khoi from UNC Chapel Hill. I would like to present our work on longitudinal atlases of cortical and subcortical tissue microstructure of the developing brain. A majority of neurodevelopment researches focus on adult brain or lifespan development. Here from a lifespan study shown on the left, we can see the cortical and subcortical gray matter volume changes over time from birth to 80 years old. We can see that the trajectory changes rapidly during the first couple years of life before stabilize around 18 to 20 years old. Similarly, on the right, by tracing the cortical thickness and myelin level from birth to 100 years old, we can also see the change is very fast and dynamic for early age with rapid changes in the trend as well as the rate of change or velocity. This suggests that early age is a very critical period of neurodevelopment where there are a lot of actions going on in the brain, including neuron genesis, axon genesis, or synaptic pruning. By charting the brain changes during this early age period, we can see how the brain develops. On the left are the volumetric changes of anisotropy and fiber coherence map from 0 to 24 months. We can see the development of the forceps major, the forceps minor, the cortical spinal tract, the internal and the external capsule. There is a development trend from center toward the cortex, from posterior toward anterior, and from inferior to superior direction. On the right are the cortical neurite and the cortical soma density changes over the first 2 years of life. We can clearly see the pattern of the somatosensory, the motor, and the visual cortex, which are in charge of very crucial early brain function. However, the study of early age neurodevelopment is challenging due to a lack of a spatial reference or atlas. While there are multiple adult brain atlas, dense temporal baby brain atlas are very limited. Most, if not all of them, are based on structural MRI data. Presented here on the right is a dense temporal atlas for the first 2 years of life using T1 weighted and T2 weighted images. Our goal in this study is to provide a community of dense longitudinal microstructure brain atlases from birth to 5 years of age. Our atlases are based on the microstructure index calculated from diffusion MRI, and thus we provide unique and complementary information to other T1 weighted or T2 weighted atlases. From our atlases, one can chart the developmental trajectory of anisotropy, cell size, tissue volume fraction, or membrane permeability. These can be used to identify typical brain development or to detect abnormal development. We use the structural and diffusional data from the Baby Connectome project. Briefly, the data were acquired using an accelerated longitudinal scheme. T1 weighted and T2 weighted have 0.8 mm isotropic resolution, while diffusion data have 6 shells with a total of 151 volumes, 1.5 mm isotropic resolution. We use the PCB preprocessing pipeline, including QC, school stripping, eddy current, and distortion correction. After QC, we use a total of 382 scans from 217 subjects. Then, we fit a diffusion microstructure model to calculate microstructure index. Our model is called microstructure fingerprinting SMSI and is based on the SMSI framework. Briefly, we simulate a large collection of signal fingerprints from different microenvironments, including axon or soma with a variety of radius, membrane permeability, or different extracellular environment. Then we find the contribution or the volume fraction of each microenvironment to the total signal at each voxel. Here on the left, we show some example of structure we use to simulate the fingerprint signal. All of the fitting were done in subject-native diffusion space. On the right are some example of the microstructure index map from a healthy adult from SCB data. From left to right, briefly, there is an intra-axonal volume fraction highlighting the axon or white matter part, The extracellular volume fraction highlight the extracellular space. The intrasomal volume fraction highlight the cortical gray matter. The free water volume fraction show the cortical CSF and the ventricle. On the map show the radius R or the permeability K. Then to construct the atlas, we build a set of 8 specific templates using T1 weighted and T2 weighted images. On the left are examples of the template at 0, 12, and 24 months. Then we bring the nifty diffusion index map to the template through the nifty structural space. From that, we do outlier rejection and averaging to get the volumetric atlases. We calculate the atlas for each month in the first 24 months and then every 12 months up to 5 years. We can see the development over time, especially the growth of the axon, highlighted by the increase in anisotropy and intra-axon volume fraction. The radius map and the permeability map here are shown using a 2D color map, where color denotes the radius or permeability value, and brightness denotes the associated axon or soma volume fraction. We plotted the general trend in gray and white matter. Overall, our results indicate increasing anisotropy and axonal volume fraction, shown by the increase of FA, MAI, intra-axonal volume fraction, especially in the white matter, and a decrease of free water content, shown by the decreasing of MD and free water volume fraction and extracellular space. This supports the proliferation development theory of continuous growth of axon and soma bodies during early ages. Also, by looking at the orientation coherence index or OCI, we can see OCI decline indicate the increasing of complexity in fiber configuration with greater orientation dispersion. Except for the first 3 months, the radius and the permeability generally remain stable, with a slight increase in soma radius and axon membrane permeability in the white matter, suggesting the axonal growth and myelination. To construct the cortical surface atlases, we map the index onto the cortical surface. And for each vertex, we perform outlier rejection and JMM curve fitting, giving a vertex-wise longitudinal curve. Again, we only show here some example time point, but our atlas is monthly up to 24 months and then yearly up to 5 years. The general trend of the cortical development agree well with the volumetric atlases. Thus, there is an increase in anisotropy and axonal volume fraction, and a decrease in water content. Notably, we can see the spatial pattern vary rapidly and then stabilize after 6 months. This pattern highlights the somatosensory, the motor, and the visual cortex. Comparing with the human connectome project multimodal parcellation in the last row, we can see some similarity and dissimilarity. This underscores the importance of a baby-centric atlas, as the adult atlas are not fully representative for the early developing brain. We hope that our atlases will help the community in studying normal growth and early detection of abnormal development. Thank you for your attention. Feel free to contact us should you have any questions.